Welcome, everyone. I'm Barry Sardis. I'm now the president of the VTE, the Villages Technology Explorers Club. Everything we seem to do these days is related to COVID, and that includes the activities of the VTE. Our first meeting of the year, seems like years ago, and because it was in person, was on longevity science. And while at the time we didn't have COVID, it turned out that there is a relationship. We had planned on having an expert on longevity science for our next event. It didn't happen. However, that expert participated in a conference called Longevity 2020. It actually was kind of a mega webinar. It was held in April. There were about 1,200, give or take, people involved in this thing, mostly attendees, of course. It was on an international scale, and it lasted for 5 days. And I attended, and the point that I'm trying to get at is that a lot of these attendees, speakers, actually were commenting on the fact that their activities are now turned over to COVID, since age-related disease and COVID are both affecting the immune system. Our second and first virtual event of the year was about Kiki, the intelligent robot companion. And Mita Yun, who is the founder, in her presentation, talked about how products such as Kiki help the elderly with loneliness by providing companionship and connections to family members. And of course, we're all aware of the situation that's happened with COVID exacerbating that problem. And even our previous event, which was Voyage taxi service, robo-taxi service, included a comment by Oliver Cameron about a disinfectant system that they're installing in the vehicles so that when someone is done with a ride, the automatic disinfection occurs, ready for the next person. So again, COVID related. And so finally, we're going to get to take on COVID head on. And I'm going to introduce our speaker today, Renee Tobias, and I have a bio that I will read. So Renee Tobias is a master's level molecular biologist with 20 years of experience in the pharmaceutical and life science industries. She spent the first part of her career doing drug discovery research at companies including Amgen, Bayer, and Schering-Plough, and eventually moved away from the lab into more commercial life sciences roles. As an application scientist turned strategic marketer, she's developed, commercialized, and supported a variety of complex analytical products for biopharmaceutical and vaccine research. Her collaborations with industry scientists and opinion leaders over the years have given her an in-depth view into the processes and challenges of bringing a new biologic to market, from early candidate discovery to manufacturing and regulatory approval. Renee is currently Director, Product Management at Berkeley Lights, Inc., a digital cell biology company whose platform enables and accelerates development of cell-based products and therapies. When she's not working, Renee loves to cook, read, and hike Bay Area trails with her 2 teenage children and her dog, Maggie. I expect we're going to have some very interesting information, and we'll now turn the stage over to Renee.
Thank you very much. It's really nice to be here. So I'm happy to be able to share some information and knowledge about some fairly complex aspects of this pandemic. I may go a little fast. I have a lot of slides to cover, but I will do my best not to lose you. And now I'm going to go to my slides. All right. So the title of my talk is Getting to the Other Side, the Science Behind Virus, Vaccine, and Ending a Pandemic. Oh, here we go. Sorry, just getting the clicker going. So I'm gonna basically, I've divided into 4 different topics, connected topics. The first part will talk about the coronavirus itself and its biology, how it works. Next, I'll be talking about the immune system response and what happens to our bodies when it gets infected. Then I'll be talking about coronavirus vaccines, their development, and how that's going. And then I'm going to talk about really what it looks like needs to happen in order to really get us through this pandemic and end it. So I will begin with the virus. Not going to spend too much time here because we all know that it's bad, that there are a lot of people who have died. There are an incredible number of cases. It's estimated somewhere between 2% and 5% of the population has been infected already. And the United States is approaching what looks like a third peak of viral cases right now. Some facts about the COVID-19 illness. We know that it emerged first in Wuhan, China. It's a virus that jumped across species. Viruses that develop a mutation that enables them to infect another species tend to be very dangerous. We've seen this before. The COVID-19 illness spreads from person to person through respiratory droplets, coughing, sneezing, talking. Most people infected have mild symptoms and recover just fine, but people with underlying conditions, elderly people, sometimes healthy people as well, can develop very serious illness. An interesting thing about this virus is that it doesn't follow a spreading pattern similar to a seasonal flu, which is really like, you say one person infects one other person or infects two other people, and it's sort of a linear spread. This virus appears to have a super spreader pattern, which means that most infected people won't actually give the virus to somebody, but there are a few people here and there who, for whatever reason, tend to shed a lot of particles and will infect a lot of people all in one place. That's the pattern that we've been seeing, and it makes the tracing and the tracking of it, the strategy needs to be a bit different. Just to clarify some terminology, first of all, this is a really cool picture taken at a university in China of the coronavirus with its spike proteins. So the term COVID-19 is used when we're talking about the disease itself. COVID-19 disease is caused by the virus that's called SARS-CoV-2. And SARS-CoV-2 is part of a family of what's called RNA viruses called coronaviruses. So coronaviruses are a class of virus. These commonly infect animals, but have been known to jump across species. Coronaviruses are named for these spiky projections that you can see in this picture on the outer surface. Those projections are, they're known as the spike proteins. So there's 7 coronaviruses that have been known to infect humans. So basically 4 of them are basically your common cold. They cause mild upper respiratory infections. But 3 of them have caused global pandemics, including the current SARS-CoV-2. So the SARS pandemic and the MERS pandemic back a number of years ago were both coronavirus. So they are COVID-19 is, sorry, SARS-CoV-2 is related to SARS, has a lot of things in common. So before I dive into more detailed biology of the virus, I wanted to put this up because I'm going to be talking about RNA and proteins and things to make sure you sort of understand how they're connected. So DNA is basically, it contains the genetic code. So think of it, if you're into technology, as like where the data is stored. RNA is a molecule that is made from that code of DNA. So DNA is read to make RNA, which is like an opposite readout of the DNA. It's like a transition molecule that is used to make proteins. So proteins are the actual execution of the code, and proteins are really the functional pieces in cells. Proteins have three-dimensional structure that dictates their function. They bind, they interact, they catalyze. They basically are responsible for most of the workings in our body is proteins. All right, so what exactly is a virus? It's a good question. People actually argue a lot about whether viruses are living or not. We know they're very small. This is a scale where you can see a DNA molecule here, 10 nanometers. So it's about 10 times the diameter of a DNA molecule. Bacteria, 10 times bigger, and then our own cells are much, much bigger. So viruses are very tiny, microscopic things. They are infectious, and they can only reproduce themselves inside a host organism. So they don't have any of their own machinery to replicate themselves. They are made up of genetic material. So they have the code that encodes themselves, but they don't have any of the other things that are needed to make new viruses. So they contain their genetic material either in the form of DNA or in the form of RNA that encodes all of those protein building blocks to make more of themselves. They also have what's called a capsid or a capsule that surrounds the genetic material made of specialized proteins, and then an outer envelope made out of fatty lipid molecules with proteins embedded in them. So the important takeaway from this is the virus has its genetic material and it has proteins on its outside surface. So how does a virus survive? So the life cycle of the virus is all about replication. So its one and only desire in life, if it's alive, is to reproduce itself and make more virus. It's not interested in killing you. It's not interested in making you sick. It just wants to replicate. So, but it needs your cell machinery in order to do so. So the way it replicates itself is by attaching to and entering a host cell and effectively hijacking its molecular machinery. So it takes, all of the different proteins and different elements to turn DNA into protein and puts it on its own genetic code and manufactures itself with the cell's molecular machinery. And you can see that I like this drawing here. So the virus attaches, it enters the cell, it injects its genetic material, makes more of itself. It assembles all the pieces into more particles, and then it escapes the cell, either by basically blowing it up or by budding away, taking part of the membrane with it that forms the envelope. But you can imagine this process of escaping the cell can destroy or badly damage the cell. And that's how viruses do their bad work inside of you and make you sick. And this is a cycle. So you release, thousands and thousands of particles from a cell, and they all go out and attach and infect other cells, and so forth and so forth. And it continues to multiply. So viruses are very specific to their hosts. They can only infect a certain species, and they can only infect a certain type of cell within that host. And the reason that has to do with the proteins on the surface of the virus and the proteins on the surface of the host cell. And those proteins are called, the proteins on the cell are called receptors. So another basic to teach you is about receptors and ligands. So a receptor protein on the surface of a cell recognizes and attaches to a target molecule called a ligand, kind of like a lock and a key. So this is a very specific interaction. A receptor will bind to a specific ligand. When that happens, when receptors bind ligands, it usually triggers a chemical signaling cascade within the cell and leads to some sort of biological response. So I think I have a drawing. Yes, there you go. So receptor on the surface of the cell, ligand is caught by the receptor, and then it triggers this cascade and something happens. So an example of this would be, for example, if when insulin binds the insulin receptor on the outside of a fat cell in your body, it stimulates that cell to start taking up glucose. So that's an example of a receptor ligand signaling. Receptor ligand interactions are the primary way our cells communicate with each other and respond to the environment. That's very, very important. But viruses are pretty sneaky because they actually use the cell's receptors in order to enter the cell. Virus proteins on the outside, on the envelope or the outer coating of the virus, will essentially mimic a receptor's ligand. So the sequence or the folding will look just like the key that's supposed to fit into that cell's lock. So the host cell receptor binds to the viral protein instead of its own ligand, and that enables the virus to enter the cell. In the case of SARS-CoV-2, that spike protein that you saw in that initial picture mimics a ligand to a receptor called ACE2. So this is the sort of just the cartoon of the anatomy of SARS-CoV-2, where you have the genetic material inside, you have the envelope, and you have proteins embedded in the envelope, and then you have these unique spike proteins. So the spike proteins are recognized by this ACE2 receptor, sticks to them, and then is basically folded into the cell. I'm hearing someone typing, so I'm wondering if people need to mute themselves. All right, so the thing about the ACE2 receptor is it's pretty much spread throughout the body. So the ACE2 receptor basically gives the virus access to your bloodstream and the cardiovascular system. And this is one thing that is so frightening about it. So ACE2 is actually an enzyme, which is a certain type of protein on the surface of endothelial cells. Endothelial cells line the inside of our blood vessels, and they also line organs like our lungs, arteries, blood vessels, our heart, kidneys, intestines, and they're even found in our nose. So all of these different parts of our body have this ACE2 receptor displayed on the outside of those cells. Endothelial cells form the barrier between the blood, the very small capillaries in your blood, and the organs that the blood supplies. So they control movements of fluids, of nutrients in and out of cells and in and out of your organs. So it's a very pivotal receptor. And once SARS-CoV-2 established itself through ACE2 receptors found in endothelial cells of the lungs, it can very easily move to other parts of the body. All right, so that's the basics of the virus. And now I'm going to shift to the body's response to the virus and talk a bit about the immune response. So you have an infection, the virus has established itself in your body. What happens? Excuse me. So your immune response comes in two stages. When we have an infection or an invasion by a foreign body. And those two stages are called innate immune response and the adaptive immune response. Both responses involve the activation and recruitment of many specialized white blood cells. Some are called lymphocytes. It's a very, very complex web of signaling and cell differentiation and activities. And I'm going to just simplify it down to some very basic pieces so that are relevant to COVID. So again, the innate immune response, excuse me, is the immediate response. And then the adaptive response is a more targeted but slower response. So there's a timeline down here to show that. So innate immunity is basically your body's first line of defense. It involves basically the physical barriers like your skin, your mucous membranes, the flora in your gut, physiological barriers, responses like body temperature. So when you get a fever, this is part of your innate immune response. The acid in your stomach is part of that defense. There's something called the complement pathway, which I'm not going to go into, but it's basically, it's like a set of toxic molecules that help kill infected cells and kill pathogens. And then there's the inflammatory response. The inflammatory response, this is like what's associated with like redness, pain, and swelling, and involves the release of protein molecules called cytokines that are very important signaling molecules. And then there's also a cellular response where certain white blood cells are activated. Many of them are called killer cells, and they actually eat the virus. They swallow it up, and I will talk about those as well a little bit later. So a lot of things happening in your body when it encounters an infection. Now here's a cartoon of a killer cell eating a pathogen. With SARS-CoV-2, something happens that's a bit unusual. As it enters different organs, tissues in your body, it's been known to trigger a very dangerous runaway inflammatory response. So part of this innate immunity that basically like throws the kitchen sink at whatever the body's discovered as a foreign invader starts to just overwhelm itself. And so this runaway response is characterized by too many of these cytokines, which are molecules that basically encourage the inflammatory response. They recruit these killer cells. They tell other cells to release more cytokines. And because the response is so huge, the body ends up starting to attack its own healthy cells instead of just infected cells. And that cellular damage leads to even more cytokine release. And it's kind of this vicious escalating cycle that's called a cytokine storm. I think we've probably all heard that term. And that cytokine storm is what is behind many of the very severe cases and deaths from COVID-19. So it's not the mechanism of virus infection itself as much as it is our body's response that's doing the damage. So the adaptive immune response is a much more specific and targeted response, but because of that, it takes a bit longer to get going. The innate immune response, the cells of the innate immune response actually trigger the onset of adapted immunity. And I'll go through that as well. It's a more robust, it's a stronger response. So instead of this general fast, like attack anything response, this is like, okay, this is the thing that we have to attack and we're gonna go get it. It involves the formation of millions of lymphocytes. These are called T cells and B cells. So these are white blood cells that are armed against the virus. So millions of these are made to help rid the body of an infection. So you have an innate response early on that triggers the activation or the arming of the T cells and the B cells. So T cells, T cells are responsible for what's called cell-mediated immunity, and B cells are what are responsible for what's known as humoral immunity, which is antibodies. So one important thing about T cells and B cells is they have to be activated against the virus. They start out as naive cells in the bone marrow. They're called naive because they haven't been activated, but they're basically hold a library of different receptor conformations, different receptor combinations called TCRs and BCRs. That's for T cell receptors and B cell receptors. So proteins on the surface that bind to ligands. BCRs and TCRs are really super diverse. So there's millions of different iterations of them. They're created by random DNA shuffling. And it's known, all these sequences together is what's known as the repertoire. So people talk about like the antibody repertoire. It's the variety of different sequences of receptors or antibodies that can be made. So each cell carries its own unique receptor. So one sequence per cell. So each cell is designed to recognize its own specific ligand. So the naive T cells and B cells, what they do is they just hang around, move through your lymphatic system, looking out for the key that fits their receptor, just in case. So this is where it starts to get complicated. So cells called antigen presenting cells are what activate the T cells and B cells and direct them to the right pathogen. So antigen presenting cells are these virus eating cells that I'd mentioned before. They're also called phagocytes. So these cells actually, they engulf and they ingest the virus by a process called phagocytosis. Here's a little APC eating something. Then once it's inside, they break up the virus's proteins into small pieces. And these small pieces are known as antigens. And they display those antigens on their membrane. So they send them all out to their surface and show them off on the outside. Now, when a naive T cell or B cell comes across one of these antigen presenting cells that happens to have the right key to fit its lock, they connect. So antigen presenting cell, T cell or B cell, they're attached, and the APC activates the lymphocyte. And it says here, go get this guy. So I like these comics. I think they're funny. So the first thing that happens is the T cells and B cells start dividing and making lots and lots and lots of copies of themselves. Some of these copies will become killer cells, and some of these copies will become helper cells. This is in the case of the T cells. So cytotoxic T cells go around looking for infected cells, and they know they're infected because your cells will also like show off the antigens. So they'll find them and say, oh, I need to get rid of this cell because it's a problem. And it'll shoot out some toxic, or it'll pull another cell that will shoot some toxic stuff and kill the self cell. Helper cells do other things to help the immune response along. They secrete cytokines, which is one thing that recruits all kinds of things and starts other things from happening I'm not going to go into, but the helper T cells also activate other immune cells. The other type of cell that one type of cell that helper T cells activate are B cells. So don't worry about this diagram. I'm not going to go through it, but just basically T cell tells the B cell, okay, B cell shows the antigen to T cell. T cell says, yes, you should go get it. B cell becomes something called a plasma cell, and that makes antibodies. Plasma cell is basically an activated B cell. So activated B cells make antibodies. So T cells are doing cell-based things. B cells are sending antibodies to go fight. Antibodies, I like to think of them as the ninjas of the immune system. They're little fighters. So they're this Y-shaped protein made of some different pieces that are attached together called an immunoglobulin, and they circulate in the blood after they've been secreted by a plasma cell. They're basically pretty close to what the B cell receptor was originally. So antibodies are like receptors. They're not receptors, but they're like receptors in that they bind to a ligand, and that ligand happens to be an antigen. So antibody binds an antigen that it recognizes very specifically based on that whole activation sequence. So antibodies do their work by basically sticking to the antigen from the virus or foreign invader with that lock and key type mechanism. The strongest antibodies against SARS-CoV-2 are actually specific for parts of the spike protein. So these antibodies can really effectively render the virus dead by sticking all over its surface. And the other thing they can do is pull over killer cells and molecules that will kind of eat the whole complex. So this is like just a diagram of antibodies sticking to the outside of a virus. The other thing that can happen is, or the other thing that an antibody can do is prevent it from binding to ACE2. This is called neutralization, which means that infectious cycle is blocked. So the antibody cannot enter the cell. And the more antibodies stick on, the sooner it will get swallowed up and gotten rid of by the body. All right, another thing that activated T cells and B cells do that's very important is they make cells called memory cells. So there's memory T cells and memory B cells. So the reason this happens is because the cellular immunity from the T cells and the antibodies from the B cells, they don't last forever. It's very expensive, like resource expensive for the body to keep making these cells and keep making these antibodies. So once an infection is eliminated, the body backs off and stops producing these effector cells, is what they're called. And that's why when people say, oh, I don't have antibodies anymore to COVID, it's not that they're not immune, it's just that their response is lowering because the infection's gone. But they will still have memory cells. These are just like a few kind of quiet, mellow, long-lived cells that circulate around for a long, long time, like decades after an infection is eliminated. And the thing that's special about these cells is they can very efficiently and very quickly reactivate that whole response of making antibodies, making killer T cells, making helper T cells. So your body can very quickly, if a memory cell encounters this antigen, it can very quickly activate that whole adaptive response. And that's what long-term immunity is. And that's what we want a vaccine to do is to create this long-term immunity against the virus. So with that, I'm going to transition into talking about vaccines. So as I said, the goal of vaccination is to create long-term immunity, but without having to actually make you sick with an infection. So vaccines prevent disease by artificially introducing antigens into the body to stimulate that adaptive immune response. So these are antigens that your body will not have ever seen before, because ideally, well, you haven't had this disease before, which is why you're being vaccinated. So this essentially kind of tricks the immune system into thinking it's encountering a virus or some other type of infection, and it triggers the memory cells to start producing antibodies. Oh, sorry, it doesn't trigger memory cells. It triggers the immune response so that it will start producing antibodies and ultimately memory cells. So traditionally, vaccines have been made from the actual pathogen itself, or it gets broken up and the antigenic parts are used. They're injected into the body and recognized by those elements of the immune system. So some examples would be the polio vaccine, which is a very, very effective vaccine that's made from the whole virus that's been killed, either chemically or by heating, so that it can't infect or replicate anymore, but it still has those proteins that act as antigens in the body. Split virus is basically a chalked up virus, same idea. So most of the influenza vaccines are split virus, rabies, hepatitis A. There's also something called a subunit vaccine, which is an even smaller part of the virus. So this would be, for example, for COVID, it would be just the spike protein. So they would take the virus, purify away that spike protein, and use that to formulate the vaccine. So subunit vaccine examples are the pertussis, whooping cough, hepatitis B, and human papillomavirus. And then there's something called live attenuated virus. Measles and chickenpox are examples of this. So a live attenuated virus is a weakened form of the virus. So they basically, it's basically grown in cells and what's called passage. So it's grown through many, many, many generations until it loses its potency. So it can no longer make you ill, but it can still replicate itself and act like a virus in those other ways and trigger an immune response. So sometimes there's concerns with these, sort of whole virus, live attenuated virus vaccinations, because live attenuated can sometimes make people sick, or they can have a very strong immune response to it. So people with compromised immune systems should never get live attenuated virus. But there's new technologies out there for creating vaccines that overcome some of those problems. So recombinant DNA technology is used to make antigen vaccines. So what's called a recombinant subunit vaccine is produced through DNA biotechnology. So basically the DNA that encodes the protein antigen, such as a viral surface protein, is injected into a bacterial cell or a yeast cell or a special mammalian cell line designed for biomanufacturing. And those cells are grown up and put into a bioreactor, and they grow. And as they grow, they produce this antigen protein and spit it out into the media that they're growing in. And that protein can be purified and formulated into a vaccine. So this method of making a subunit vaccine doesn't involve any virus. So there's no need to grow up lots and lots of virus in a lab, which you can imagine in the case of SARS-CoV-2 would be extremely challenging because it's so infectious. I think it would be a biosafety level 3, which is hazmat suits, the whole thing. Very expensive, very difficult. So recombinant DNA technology provides a safer, much more efficient way to produce antigen vaccines. One setback or one disadvantage of antigen vaccines, though, is since they're proteins, they're solubilized, so they're actually dissolved in liquid, which means, well, they're very small, and it means that they're not very good at actually being immunogenic. And so because bigger things tend to be recognized much more easily, bigger insoluble things like viruses. So they're given with what's called an adjuvant, which is a chemical additive that helps to actually like wake up the immune system and say, hey, look, there's an antigen here. And so adjuvants can be aluminum hydroxide is one that's been used for a long time. Some nanoparticles, oil emulsions, things like that are part of these formulations. So it's also something to think about. But there's a new generation of vaccines that are being developed and can be developed much more quickly. Viral vector vaccines and DNA vaccines and RNA vaccines are basically made just with sequence information. So they're very fast to develop. Just having the sequence is enough to start a project. You don't have to have anything else. And synthesizing DNA or RNA is much, much easier than it used to be. So the advantage of these types of vaccines, a big one is that they're very adaptable. So programs can move very quickly. So Moderna is a company that's really on the leading edge of the vaccine race. It took them 42 days from the sequence analysis of the virus to create a new vaccine, their candidate, which is mRNA 1273. 42 days for something that normally takes years. It's very exciting. This is just a schematic of how viral vector vaccines work. I'm not going to go through the whole thing, but the concept is that a harmless virus, a different harmless virus is used to generate the immune response. So the DNA encoding the antigen is packaged into often what's an adenovirus, which is a virus often used in biotechnology that does not infect people or make people sick. So that version of the virus is injected in, it encodes an antigen, the antigen is presented and stimulates the immune response. So no virus, no pathogenic virus needed. It's just this harmless tool that's used. RNA vaccines skip it all. So basically an RNA vaccine is just a direct injection of the RNA into your body where it enters an antigen presenting cell, which makes the protein from the RNA and then presents it on its surface, triggering that activation cascade that I had talked about. It's very cool. They do, so mRNA is not a very stable molecule, and mRNA is just, it's another word for RNA, it's messenger RNA, it's not important, but RNA vaccines use nanoparticles for delivery. So often, I think the Moderna one is a lipid nanoparticle. So it's just like a coating that encapsulates the RNA and protects it from being degraded once it's in the body. And it's a lipid particle because that's what cell membranes are made of. So it actually can just fuse with the cell membrane. They're working on an oral one that will just fuse with cells in your small intestines. It's quite nice. And this just shows some different types of nanoparticles that are being used for pharmaceuticals and vaccines. So packaged RNA. All right, how's the vaccine developed? Well, there's this initial stage of making it, right? So you're either growing up virus, you're purifying antigen, you're synthesizing your RNA, all of those pieces happen in that exploratory development piece. And once you have something that appears to be a candidate, it starts to go into clinical testing. So the preclinical stage is all about safety and toxicity. So they give the vaccine, and activity. They give the vaccine to animals, like mice or monkeys usually, just to see if it actually triggers an immune response. Phase 1, so the clinical testing is in 3 phases, the next stage. So phase 1, the vaccine's given to a small group of maybe 10 or 20 people just to determine that it's safe and to study the response that it triggers. Phase 2 is scaled up a bit to hundreds of people so that they can kind of explore dosage, do more exploration of safety and immune response. Phase 3 is really the really important one. So phase 3 is scale up to tens of thousands of people. I think 30,000 people is the requirement. That confirms its safety. It gives you the opportunity to see if there are any rare side effects or illnesses. And it also can give you an idea of its effectiveness. And the reason they give it to so many people is because it's not ethical to actually give someone the disease that they're vaccinating against. So you need to have enough incidence of people contracting the disease just normally to get a representative enough sample to see a difference, right? So 30,000 people, how many of those people are going to just in their daily lives happen to get COVID-19. It's, you know, it's a small sample, but if you only have 10 people that contract the virus, that's a very small comparison to make between people who have not gotten the vaccine. I hope that makes sense. Maybe I should diagram that out, but it's the sample size that matters. So it tells you whether it's really effective and confirms safety findings in a bigger population. Right now, there are 11 vaccines in phase 3 clinical trials for COVID-19, which is very exciting. Some of the technologies I told you about are really accelerating that development in combination with just the push to authorize by the FDA. There's a political side to it, we know, but as long as those vaccines get through those phase 3 clinical trials with the right outcome, it can be considered safe and effective. So this is just a table of what's in phase 3 right now. So there's some inactivated or killed virus, like the polio vaccine. There's some a recombinant subunit, the viral vector vaccines that use adenovirus, including the Oxford one where that schematic was from. And then there's a couple of mRNA vaccines, including Moderna, so Moderna and Pfizer. Those are ones to keep an eye on, as well as the AstraZeneca and I think GSK here. No, not yet. So anyways. All right, so that's vaccines. How am I on time? Oh, sorry, I'm going to run a little over. All right, I'll go fast. So this is the last section where I just wanted to kind of talk more about like the epidemiology and what's really needs to happen. So our best bet for getting back to normal, what we want to achieve is herd immunity. What's herd immunity? Everybody talks about it. Herd immunity is protection for everyone and for the vulnerable without everyone having to be sick. So because of the way the virus life cycle works, if a virus doesn't have a new host to infect, it can't spread, which means it dies. So if enough people are immune, it doesn't have to be everybody, but if enough people are immune, the chances of the virus encountering another host to infect becomes much, much smaller. And you can see by this, this diagram here, if most people are immune, not everyone has to be immune, because these people who are not immune are not very likely to come into contact with someone who is infected. And even if they do, it's harder to get past the next step and the next step. So it essentially stops the spread of the virus. No more new cases. So we can get to herd immunity by two different possible paths. The first one is a vaccine. So that is what a vaccine can do, is it can achieve this herd immunity without everyone getting sick. And the other route is just through natural infection, which is letting the virus infect who it will until enough people become immune that it stops spreading. But this is a strategy that it's been terrifying to me to hear it talked about, because it would be a disaster. And the reasons are, it's not like the flu, right? Many people infected with the virus die from it. It's some places I've read say 3%, it may be 1%. I think the CDC is using 0.65% in its figures. That's a lot of people. Plus, the people who don't die, many of them get serious long-term illness. And you've seen now, because of the mechanism of the viral infection, how it can access the body and cause damage to organs like your kidneys, your lungs, your heart, etc., because of this cytokine storm. The fatality rate is believed to be somewhere between 1% and 3%. So if we went to herd immunity, which would require, according to the World Health Organization, about 70% of the population to be immune, that would mean over 2 million Americans would die before things slowed down. And imagine the stress on our hospital systems, our healthcare would be overwhelming. And then the last piece of this is that it's one thing to talk about herd immunity when there's not an active spread of disease, right? Where we give people a vaccine for the measles and maintain herd immunity. This is an ongoing pandemic, which means it will reach that theoretical threshold of 70% of people being immune, but it's not going to just stop like a light switch, because there will be so many infected people that the virus will continue to spread, meaning that probably more like 90% of people will need to get the virus before it actually stops. And then lastly, it's impossible to isolate vulnerable people in our population because they're not all in one place. They walk among us, people with diabetes, people with asthma. So anyways, all right, so based on that, we want a vaccine. We want the vaccine route to be the way we achieve herd immunity. So how effective does the vaccine need to be to get us there? And this is a really big question. So a vaccine's efficacy is defined by the percent reduction in infections. So how many infections it prevents is its efficacy. So in other words, if 100 people who haven't ever seen the virus are given this vaccine that has an efficacy of 80%, on average, 80 of them would be immune. They would not get infected. But effectiveness of the vaccine refers to how it acts in the real world, and that is usually lower because not everybody gets the vaccine. And it also depends on how many active infections there are. So this is a great chart from the American Journal of Preventive Medicine from a study that was done. This is a computer simulation looking at potential coronavirus vaccination. So they modeled vaccine effectiveness based on the simulation of 330 million Americans all interacting with each other normally. And there's a lot going on here. So each of these lines represents a situation where a certain percentage of the population actually gets the vaccine. So in this red line follows what happens if everybody gets the vaccine, down to this line, which is if only half of people get the vaccine. This axis shows the percent reduction in cases, which is its efficacy. And then this line shows how effective it is. Oh, sorry, this axis refers to the efficacy. So in other words, a vaccine that is 60% effective, or let's go to the top. So in order for 100% of cases, in order to have a 100% reduction in cases, which means it stops the pandemic, we need either a vaccine that's 60% effective and 100% of the population gets it, or it needs to be 100% in efficacy if only 50% of the population get it. So you can see how this really trends down. So only some of the population getting the vaccine really affects how well it can stop the pandemic. And this gets even worse if more people are infected. So this is the same model, assuming that 15% of the population has been infected with the virus. So the first one, this is if 0%, close to 0%, I think it was actually 1% of the population got the virus. And this is if 15% of the population got the virus when the vaccination happened. So there's no reaching 100%. This is why it's so important to slow the spread too. So I hope those are somewhat clear and I can answer questions later. Okay, so that shows us how effective it needs to be, but how effective will it be? So right now the FDA is requiring a released COVID-19 vaccine to be at least 50% effective in protecting against the illness. And it's likely that the first generation vaccine will probably be 50% to 60% effective, which is about what the flu shot is. The flu shot's like 40 to 60%. But the bad news is the latest polls are showing only 60% of Americans say they want the vaccine. So if we go back to our chart, so 60%, so somewhere in between this line of a 60% effective vaccine means you still have a pandemic. It's slower, but it hasn't defeated it. So we need everyone to get that vaccine, or cases will likely continue to rise. So it's bad news, but don't despair, because it's not our only weapon. COVID-19 vaccine may not be the magic bullet, but it also won't be useless. So even a vaccine that's only 60% effective will very likely mitigate the illness. So there will be fewer severe cases, fewer deaths, and symptoms will be milder. If we prioritize vaccinating vulnerable people, we will save many, many, many lives. That's a good thing. And then there are other interventions that we have that will contribute to stamping it out. Ending the pandemic will take time and will require a combination of approaches. So vaccine is not going to be the only thing. Non-pharmaceutical interventions, number one. These are those things like the social distancing, wearing masks, staying home when you can, etc. These are the interventions that slow the spread of the disease in the community. So they minimize the number of infected people. They keep our healthcare system from being overwhelmed, and will also make it help the vaccine be more effective. Novel therapeutics are being developed, particularly monoclonal antibodies engineered in the lab. These are neutralizing antibodies that typically will bind that spike protein and prevent the virus from entering the cell. These can offer some level of protection until a vaccine arrives. So they're very good for treating early infections. They're more effective and they're safer than the repurposed drugs that are being used now, like the remdesivir, the hydrochloroquine, the methotrexazone. I think that's how you say it, but anyways. And they're very targeted therapies. They can protect people who are already sick or whose bodies can't mount strong immune responses. And after they're in your system, they can potentially last for months. So this is like the cocktail that President Trump had. And they will often have 2 antibodies or 3 different antibodies in a formulation because they bind to different places on the spike protein. Herd immunity, that's our big tool, achieved probably through a few iterations of vaccines. So the first one won't be super effective, but it will be improved upon over time. So get vaccinated. And then lastly, there's this phenomenon that happens with viruses because they're constantly replicating themselves, which means they're always making new copies of their genetic material. And when that happens, a lot of mistakes happen. And those mistakes are called mutations. Some mutations are not viable, but some, the virus will change, which sounds really scary, but the selective pressure on the virus, remember, is to not kill its host. So it's much better for the virus to keep us alive because it can keep reproducing. So if a virus kills its host really, really fast, it's not going to get very far. So the selective pressure is to be more infectious, which means it can get in and attach better, but not to be more deadly. So over time, viruses tend to become less potent. And this is what happened with the 1918 pandemic eventually. And the flu viruses that we get today are, well, indirectly, but they're basically descended from that 1918 influenza. And it's, it's severe for some people, but it's nothing like the disease back in 1918. So coronaviruses are different from influenza. They don't mutate as quickly, but there's reason to believe that this could happen. New technologies are coming up that are really speeding up development of biologics, antibodies, and vaccines. There's this video. I don't think I have time to show it to you. It's actually about the company I work for and the product that we make, which is able to isolate single cells at a very large scale. And it's a tool for really speeding up development. So I guess I'll let you all watch that on your own time. Moral of the story is we all need to be patient. We need to follow the CDC guidelines, and we need to trust the vaccine. The clinical trials prove safety and efficacy, so get vaccinated. And that's the website to the CDC, which is chock full of information. And that's it.
Well, thank you very much. We have a few questions. Can you hear me okay?
Yep.
Okay. There's a few questions regarding mutations. First of all, what's the chance of COVID-19 mutating into an even more deadly virus? You kind of addressed that. But generally about mutation, can it make things worse? Will it require new vaccines to be made and so forth?
Yeah, that's a great question. So the SARS-CoV-2 went through, or SARS-CoV-2 did mutate, and that mutation was what enabled it to jump across from bats, we think bats, to people. And then there's potentially another mutation that happened that made the virus more infectious to people, meaning it can spread, it can attach much more tightly to the ACE2 receptor. So more virus can get in if you happen to be infected. So I would say those mutations definitely made it worse. When you think about the selective pressure, and just to caveat, my knowledge of this is not terribly deep, but my basic understanding is that the selective pressure on the virus is not to become more deadly. It will mutate, and yes, we will probably need to update the vaccine. I know it doesn't mutate as much as the flu, which we need to redevelop every year. So I really don't know, but I would say it's unlikely that it will mutate to become a more deadly virus.
Well, good, thanks. Another question came in from Peter Holmes about people who've gotten the COVID virus twice. So it looks like maybe immunity doesn't happen.
Yeah, that's an interesting one. I was following that a little while ago, but I haven't lately. It's very rare that that happens. And I know in some of those cases, the second infection is much milder, but then there have been cases where it's been worse. And again, those are extremely rare. And, you know, that could be a function of that person really not being able to mount a very strong immune response. That happens, especially as you age. your ability to have that robust adaptive response weakens. So yeah, so it's definitely possible, but I think for most of us, we probably don't need to worry about that. I think it's focusing on that vaccine, keeping ourselves safe, and getting that initial immune response is the most important thing. And the most likely scenario, even if it can reinfect, is that the next time it will be a much milder illness.
Good. Another question came in, a quantitative question. What's the percentage infected now? And so what percent efficacy is needed in a vaccine? And related to that, what's the efficacy of masks as well? And how does that play into the equation?
So I'll tell you what I know. So the number of people infected is estimated to be, I've seen a 1% figure, and I've seen a 5% figure. So it's definitely less than 10%. So we're still in good shape for a vaccine. So a vaccine that's like 60, 70% effective, it will do a lot of good as long as people get it. That's really the critical thing, is everybody needs to get it. Masks are interesting. So the rule of thumb with masks is, so if a mask helps a lot. A mask basically traps those larger respiratory droplets that are emitted when we talk and sneeze and cough, and they go really far, really far. So a mask holds those in and keeps them close to you, so that combination of masks and being 6 feet away from somebody means you're basically not exposing them at all. Masks are very good. Masks should be at least 2 layers of tightly woven cotton material, or they should be disposable surgical masks. They should not be scarves or bandanas, or gaiters. And if you can blow out a candle when you have the mask on your face, it's not very effective. That's the rule of thumb. And if you can't breathe through it, you should be able to breathe through it. It's not working very well either, because you're basically sucking, the air is going to go through that path of least resistance. So the air won't actually filter through the mask. It will just come through all the gaps in your face. And if you're using an N95 mask, it should be sealed tightly on your face for that reason, because the pores are so small that the air will resist going through them. So you want to prevent those gaps. When medical professionals use N95 masks, they have them fitted to their face. So keep that in mind. So the best thing is either those disposable surgical masks or a 2 or 3 layer cotton mask that you can breathe through but can't blow a candle through. Hope that answers your question.
Very good, thanks. Another question came in: the biotechnology producing antigen sounds promising, and how effective is it compared to traditional vaccines? And what are some successful examples of next-gen vaccines used today?
So let me think. I don't have my notes, the notes on that in front of me. So the recombinant subunit vaccines are very safe. The challenge with those is their effectiveness, because they require this use of adjuvants. With that said, adjuvant technology has also come a very long way, and there are a lot of new substances out there that seem to be very good at eliciting that immune response. So it's a very good way to make a vaccine. Some flu viruses are recombinant subunit vaccines. I think it's Novavax that has a COVID one in development, and I can't think of offhand of any on the market. Next gen vaccines, so RNA vaccines, there are no approved ones yet. And vector vaccines, there may be 1 or 2, but I'd have to look that up. Maybe I can follow up with that person about the vector vaccines.
Okay, another question. Israel is starting this week to test a new vaccine that works differently from those in the US. Are you familiar with it? Do you have any comment about it?
I don't. Do you know what kind? Maybe I can Google it really quick and see.
Tony says he has some information on that, so we can hold off on that one if you'd like.
Okay, cool.
So even if the virus doesn't have a more deadly mutation, is there another mutation that would elongate the pandemic, making it a longer term problem in general?
Well, I think that one has already happened, and that's the spike protein. There's an amino acid substitution that basically helps it attach and enter the cell much better. It's for a mutation to take over, there has to be some sort of selective pressure. So mutations will happen all the time. They're random. Most of them will have no effect. It has to be a change that affects the way the protein is folded, that adds to its functionality. And those things happen pretty rarely. And there's also a limitation to how many possible substitutions can happen that will really have an effect. So my guess is that we've got the bad one has already taken over the viral population. And, again, I'm no expert at all, but I think that based on the knowledge that I have, that it's unlikely. And the more likely scenario is that eventually it will weaken.
Okay, just have a couple.
That's not a satisfying answer.
A couple more questions for you. One is, there was an early study that concluded that people with a negative blood type were infected, were more likely to develop life-threatening cases. Has this held up over time because the person asking is and his wife both have a negative?
I don't know a lot about that or the pathway involved in that. So I would like to do a little research on that, see what the mechanism is, if it's been worked out, and then get back to them.
Okay, we can make that happen.
Yeah.
And one final question from Sid. What's the range of a dangerous sneeze of virus impregnated material? If someone is infected wearing a mask and 30 feet away, is that still, oh, and not wearing a mask?
Oh, not wearing a mask?
If they're 30 feet away, is that still?
Yeah, it's pretty far. I think it's like 12 to 20 feet or something.
Yeah.
And it's, so the risky place to be, is indoors with a lot of people, right? Outdoors where air is circulating, where you're removed from people, that the risk of transmitting particles is very low. When you get to an inside space with people, the air that they breathe, the particles, the aerosols that they breathe are not being carried away. They linger and remain in the room with you, depending on the ventilation, right? If there's no HEPA filtration, things are going to accumulate. If you're in a room where people aren't wearing masks, where they're talking loudly, having conversations like at a restaurant or a bar, 6 feet is not going to help.
Or 30 feet.
30 feet may not help. Again, it's time, it's number of particles that the person is shedding, or the number of particles that you're exposed to, and its distance. Those are the 3 variables that affect your risk of contracting, right? If you're in a room 5 feet away from someone who's infected and shedding particles for 5 minutes, you're going to get it. But if you change one of those variables, right? So put that person 30 feet away, 5 minutes, if they're really contagious, you're less likely to get it. You need to be thinking about all of these things. Passing someone at the grocery store when you're 2 feet away is much less risky than standing 6 feet away from them and talking for half an hour, right? So you can be close if you're not there for much time. Does that make sense? Or you can be far away for long periods of time. We don't worry about surfaces as much because the likelihood of getting enough concentration of live virus when you touch your face is pretty low, right? So that's like the concentration of, or the level of exposure, right? So it's really important to think about those things, that and being indoors.
I have a variant on the question because I spend a lot of time outdoors. So what about being downwind outdoors from somebody as opposed to upwind? I always try to be upwind from other people, so I know that that's practically 100% protection. But if you're downwind, could you be 30 feet away and somebody sneeze and all those particles come at you there?
I don't know for sure, but I personally agree with you. Airflow,
yeah,
aerosols, particles, they're going to be carried. They're going to spread as they pass you. But, when I'm running on the trail and I pass someone coming towards me who's running and breathing hard and not wearing a mask, that makes me very nervous because they're putting out all of these droplets, even though it's outside. If you're downwind, they can blow at you. So if you're downwind of someone not wearing a mask, more distance, right? Because
Yeah.
exposure, time, distance. So yeah, these are things to think about for sure.
Okay, well, we've covered all our questions, so thank you very, very much for that. I'll turn it back over to Tony. He has some additional information that he wants to share as well. So thanks very much for the presentation and the answering of the questions. It's terrific.
Yeah, we really enjoyed it. Thank you very much, Renee. There are still questions hovering in a lot of people's minds. We have one brave soul who'd like to come join us in the in this practice room, Martha Stillman. So I'm going to bring her into the room and introduce you to her. Others in the audience, if you want to hang around and listen to our sort of fairly informal conversation from here on in for just a few more minutes, you're very welcome to. But we've been at it for an hour and a half, which is a pretty long time for us old folks to be sitting on a chair. So Martha, welcome to. Let me get you to put your video on as well. Start video. I can't see a start video. Well, we'll have to pretend we can see you, Martha. Welcome. Do you have a question, or you just wanted to say hello? Martha. I can't get anything from Martha. Can you hear me, Renee?
I can hear you. Yeah.
Martha doesn't seem to be.
She's not muted.
She's not muted, but I don't hear her, and I don't see her.
Maybe she took a bio break.
Well, I apologize, Martha. The last time we did this, we had a couple of people up and it worked very smoothly. So I think at this point then, apart from those of us still in the practice room, it's time to call the meeting to an end, go exercise, get some blood flowing and keep away from other people and wear those masks. I'm not sure about the vaccine yet because that's still much up in the air, but we can all wear masks and we can all socially distance.
Okay. Tony, can you hear me?
Martha, is that you?
Yeah, I solved my mic problem, but I just want to say, you answered so many questions from the Q&A that you've questioned me out, that's for sure.
Oh, I'm so glad.
Yeah, I feel the same. We've really. I've got your video here, Martha. You can just join us and say. Hello.
Yeah, I'm gonna stay behind the curtain.
All right, don't be shy. Okay, then I think we've beaten it into the ground. Thank you very much. If our friends on stage would like to hang around just for a few minutes, we can have a debriefing. We kindly ask everybody else to pack their bags and go enjoy the rest of the afternoon. And thank you all for coming.
Bye. Thank you.
So Roberta, can I ask you one question? Why not?
We're listening, Martha.
Oh, okay, sorry. I didn't see a nod, so I didn't know if she heard me. So this is a just curious question. When Trump got sick, and believe me, I don't care about Trump. I care about if I get sick, what lessons can I take away from that? And one thing that's clear to me, I'm a member of the Kaiser system, is that if I get sick, I'm going to have to advocate for what I think is the best treatment. I can't just be passive and say, oh, whatever you think is best.
Yeah.
And from what I understand of Trump's treatment, the dexamethasone is probably what's responsible for his euphoria. And that's just, from what I understand, that's just cosmetic. I mean, it makes you feel great. And if you're having a cytokine storm, then it certainly is called for. But just because I'm feeling ill is no reason to ask for it. But something that's an antiviral, from what I understand, could be effective. And I could certainly imagine being in the Kaiser system, if they're short of drugs, they're not going to recommend it. And if I raise a hoot and a holler, I might be able to get my hands on it. So my question is, from what you know, are antivirals a good class of drugs to take early in an illness, even if they're not otherwise warranted?
Yeah, definitely. I think I don't, the challenging thing with this virus is that really still so little is known about it, and what kind of therapies can be effective. I mean, we're seeing, the remdesivir is not terribly effective. The hydroxychloroquine is not very effective. But yeah, antivirals need to be taken early. So at an early stage in the infection, I would imagine that could definitely help. There's other treatments now that are being investigated. Well, of course, there's the convalescent plasma option, which is, I think it would be very hard to get that early on. I think you need to be very ill to qualify for that. Heparin treatment is another one. So the virus inhibits like an anticoagulation pathway that involves regulation of heparin. So they're finding that giving people heparin prevents some of those complications involving blood clotting and things like that. What else?
Do you think that heparin is more than just an anticoagulant?
It's for coronavirus, yes.
Okay, I did not know that.
Therapy, yeah. So that's being looked at. There's something called ACE inhibitors that actually bind to the receptor and keep the virus they like compete with the virus for binding. And then they're making like decoy cells or decoy nanoparticles that have ACE2 adsorbed to them to kind of like decoy, so the virus like sticks to them instead of the receptor, but I think these things are all still being proven. So there's not really a lot of options out there in early infection. So if I could get an antiviral, I would get one. And really, I know they say that the severe illness can come on very quickly and very strongly. So definitely monitoring, and don't let them tell you not to come in if you're worried. Like if you have trouble breathing, go. Don't let them send you home, is what I would say, because there are other even non-pharmaceutical measures that they can take, like oxygen. They turn people over to help them breathe. They've been learning so many things that has prevented a lot of death. So yeah, that's what I would.
Can I ask on that topic, were you surprised that Trump turned around and is now sort of bubbling along like, I mean, I don't
Was
know.
I disappointed? Sorry.
Well, I don't want to get
Yes.
political, but he's 74 years old, and his schedule at the moment is horrendous, and not many of us could keep up with what he's doing. And it just seems quite amazing that he either recovered so well or he never had it in the first place, and it was part of a political game he was playing.
Well, he was also given that Regeneron treatment. That could very well have been part of it. I mean, he had access to care and therapies that no one else does. I mean, you can't judge a therapeutic by one anecdotal case, but it wouldn't surprise me if those cocktails of antibodies end up being extremely helpful.
We're losing 1,000 people a day. Maybe we should pick a few of those and try and help them.
Yeah. Yeah, I would like to see some of those come out. But I mean, they take a long time, and it's not just the development, it's the manufacturing them. They're proteins, and they're manufactured in living cells, and there are a lot of byproducts of those manufacturing processes that can be very dangerous to inject into your body. So there's still a lot of safety concerns. So again, those clinical trials are super important, but I think the faster they can get through them and make those things available to people, the better.
We got one other visitor from the floor, Howie Bloomstein, who's notorious for asking questions, and he's sitting here. Howie, what was your issue?
Well, I was just listening, but in your presentation, I think you're saying the more people infected, or the percent more infected, the more effective the vaccine has to be. Why is that the case? You would think the more people that are infected, there's less people around that can get it.
Yeah, it's a bit counterintuitive. The more people that are infected, the more people are spreading the virus. So when you think about, if you think about that herd immunity diagram, you need to have, so many people around you that are immune, if you're not immune, in order to be safe. And if there are a lot of infected people around you and you're not immune, you're going to catch it. So the higher percentage of the population that's actually able to infect people, the more difficult it is to obtain that herd immunity and stop the spread.
If someone's affected, over time they get over it, don't they? In other words, if someone got affected in March, April, and May, I would think by now that they're not spreading it anymore. So isn't there a
right, but the
around?
number of active infections increases with the increased spread. So the more people have gotten it, the more active infections you have until you turn that corner. So it's an indication.
I've heard with this, KCBS Radio has Ask an Expert Monday through Friday, except for holidays, around 20 after 9. And the one thing I heard that another reason for wearing masks is not only, God forbid someone has it, and they can't spread it as easily, but they're saying if you're wearing a mask and you get the infection, you get a lower dose of it, so you'll get a milder case. And that ought to be a point to make people aware that wearing a mask not only helps if someone's infected from spreading it, but if you're not infected, it's still a good idea to wear a mask even if you get it, because you get a less severe case.
Yeah, that's an excellent point. Yeah, they do believe that the viral load when you get infected has a direct impact on how severe your case is. So if you have 100 particles that all attach inside your lungs, and then they multiply and they multiply and they multiply, you're going to have many, many more virus particles much more quickly in your system versus if you have 2 or 3.
I think that's a good point, but I think we've kept you here for 2 hours now, Renee. I think we need to draw the curtain down. Thank Martha and Howie for joining us.